Macroscopic morphology with diagnostic correlation. So when we receive specimens in the lab, the first thing we're going to do is gram stain them or look at them directly under the microscope, if indicated. But the very next thing we're going to do, or the very first thing if the gram stain isn't indicated, is we're going to plate them. And we're going to put the bacteria or the specimen on different types of plates based on the specimen source. So if we have, for example, a urine culture and they suspect that the patient has a UTI or urinary tract infection, we're going to plate that on a blood agar plate, and then we're going to plate it on McConkey agar, because McConkey is a selective medium that's going to select for the growth of gram-negative organisms. And more often than not, our causative agents of UTIs is going to be a gram-negative. Now, of course, there's some exceptions, and the blood agar plate is going to capture those exceptions. So we can look at the blood agar plate to see, is on the off chance it a gram-positive, did nothing grow on the McConkey, and we can get that information that way. But based on where that specimen came from, we're then going to determine what plates to put it on. Some labs have little cheat sheets that will say if it's this type of specimen, you put it on this, this, and this. If it's this one, you put it on this, this, and this. Other ones will actually print out what plates you're supposed to use on the label. So as soon as you scan in the specimen to the computer saying that you have received the specimen and you go to look at the orders, you can see what they've ordered, and then you can print the labels. And when it prints the labels, it'll print out on the label exactly what plate you're supposed to put it on, which makes things a lot easier, takes the guesswork out of things, and ensures that everyone is operating using the same types of protocols. So when we finally incubate them, we're going to then evaluate the colonies, and that's what this whole lecture focuses on, is looking at those initial colonies on the plate. So when we look at colonies on the plate, it's known as the colony morphology. Morphology refers to like the shape, size, color, all of the physical characteristics of the colony. If you remember from microbiology class, your general microbiology class, a colony is when you have one cell of bacteria landing on an agar plate, and that one cell divides and divides and divides and divides and divides and grows into a visible clump of cells. So what was an invisible one cell on the plate, if we allow it to grow, it'll grow into a big clump. And every species grows into a clump in a different way. And that's what we look at to help give a presumptive diagnosis of what's causing the issue. We never just do one test on a microorganism and say, yep, this is what it is. We always do multiple tests. So 3 easy things we can do are a gram stain, colony morphology on different plates, and then we can follow that up with some metabolic testing. That gives us 3 pieces of evidence as to what the organism might be. There are some subtypes within different species that can do some pretty crazy things. So you think, no way is this Klebsiella, and then all of a sudden it turns out to be Klebsiella. So there's always those rare exceptions, and that's why we want to follow up with multiple ways that we're confirming the identity of the microorganism before a definitive diagnosis can be made. So, colony morphology, the characteristics and form of the bacterial colonies. We compare this with the direct gram stain smear, and distinctive patterns can distinguish some pathogens and facilitate a presumptive identification. So colony morphology provides a presumptive identification to the physician in times of critical need. So if they've been waiting a while for culture results, you can be like, it looks like this on the plate. Let's follow up with this antibiotic. Or maybe they've been waiting a while for culture results. Nothing's growing, so then they might have to pivot and think of it more of as like a viral infection. It enhances the quality of patient care through rapid results and cost effectiveness. It prevents time-consuming testing that yields few results, and helps jumpstart therapy with antibiotics. It plays a significant role in quality control, helps maintain the accuracy of commercial or automated systems, and helps to determine mixed cultures or errors in commercial systems. Initial observation and interpretation of cultures. So primary plating refers to the inoculation of clinical specimen onto the initial media. It allows for the visualization of growth and colony morphology. Observe colony morphology at 18 to 24 hours after you've cultured it. The age of the culture may affect the size and characteristics of the colonies. So here is a list of commonly used media in the clinical lab and what it's used for. Throughout this course, you will be responsible for learning very specific types of media and what it's used for, and all of the details of what's in the media and what makes it selective or differential, and all that type of stuff. So this is just kind of a cursory look up front. You do not have to memorize this list today, but as we go through the course, you're going to learn a little bit more about the different types of media, and you will be responsible for learning those details. So the initial interpretation of cultures, the initial distinctions between gram-positive and gram-negative isolates can be done using selective media. On blood agar, you can just look at morphology. It grows most organisms. It's not going to differentiate between gram-positives or gram-negatives. Chocolate agar also grows most organisms as well as the fastidious organisms. So if something doesn't grow on blood agar but does grow on chocolate agar, that can give you a hint that it may be more of a Haemophilus or a Neisseria type organism. McConkey's agar, gram-negative organisms, particularly enterics. So McConkey does select against the growth of gram-positive organisms. Differentiation using plated media. So lactose fermenters would be pink, non-lactose fermenters would be colorless. What happens is when they do ferment the lactose, they're going to absorb some of the pigment from the media. So when they do that, you may see some clearing around the growth. You want to look at the actual colonies. If the colony itself has a pink color, then it's a lactose fermenter. If the colony itself did not change color, then it's a non-lactose fermenter. Comparative analysis of the culture media is very important in the initial interpretation. So here's some different agars. This is a blood agar plate, this is a chocolate, and this is likely a McConkey agar. So McConkey agar, this is one that you do need to know the details of. And I'll add a few notes to these slides just so that you have them written out for you. All right, so a few notes have been added to the slide here. So McConkey agar contains crystal violet and bile salts, which select against gram-positive organisms. It also has a neutral red pH indicator, and it contains lactose. So it selects for the growth of gram-negative bacteria and allows us to distinguish between lactose fermenters and non-lactose fermenters. So if we have bacteria plated on here, if it's gram-positive, the crystal violet and bile salts will not let it grow. So those will die off. So only the gram-negatives are going to grow on here. For the most part, there's always a few exceptions, but for the most part, only gram-negatives are going to grow on here. These also contain, so that makes it differential, or selective, sorry. So it selects against gram-positives, meaning that it doesn't allow those gram-positives to grow. It selects for gram-negatives, meaning that it will allow the gram-negatives to grow. The next thing is it's a differential medium. So it contains lactose and that neutral red pH indicator. So if the bacterium can eat lactose, it can ferment lactose, it's going to produce that pink color. So you can see on the top, that's pink, so that's a lactose fermenter. If it cannot, it's on the bottom. You can see it doesn't change color, or it maintains the color that it would have naturally grown on any other type of media. So lactose fermenters from non-lactose fermenters can be identified here. The important thing to note is the crystal violet and bile salts make it selective. It kills off gram-positives, only allowing the gram-negatives to grow for the most part. And then the neutral red pH indicator, it contains lactose, and it helps us differentiate the lactose fermenters in pink from the non-lactose fermenters, which are colorless. So you do have to know all of those details, and it's not just for this class, it is also for that qualifying exam. They are going to ask you specific questions about the different media. So McConkey agar is probably one of the most common media that we use in the clinical lab. We get the most urine cultures sent down to the lab, and every urine culture is going to be plated on a McConkey. McConkey is typically incubated in ambient air, so we just put it in a regular oxygen-containing incubator. It contains the same concentration of gases as we would have in our normal atmospheric environment. So this shows you a picture of E. coli versus Citrobacter-like bacteria. So you can see here it's kind of far away, and here you can see it close up, what those would look like. And Klebsiella-like bacteria, so you can see far away, but then also close up. So this one you can tell looks very dry. The edges are very even, while this one looks kind of goopy and gunky and kind of messy like this. So mucoid-like colonies. So microorganisms, colony characteristics, all the laboratory scientists to make and allow the laboratory scientists to make an educated guess regarding the identification of the isolate. Colony characteristics vary among species and strains within the same genus. Hemolysis is another way that we can differentiate bacterial growth. So observation in the media immediately surrounding or underneath the colony. It's caused by enzymatic or toxin activity of the bacteria. It's most important in the presumptive identification of streptococci and enterococci. And you want to use a transillumination or a light source behind the plate to help visualize the hemolysis. So there's a light source, and then the plate, you're going to then be able to either see through or not see through it. The major types of hemolysis are alpha and beta. Alpha is a partial clearing in blood that results in a green discoloration of the medium. Example includes Streptococcus pneumoniae and certain viridin strep. Beta hemolysis is complete clearing of blood cells around the colonies. Examples would be Streptococcus pyogenes, which causes strep throat, Streptococcus agalactiae, which is a group B strep that can cause some major issues when a mother is delivering a baby. If she's positive for group B strep, that can then get into the respiratory tract of the baby and can cause meningitis and all sorts of bad stuff. So we always screen mothers for group B strep before they deliver. And Listeria monocytogenes. So Listeria, a really bad bacterium. There's been some outbreaks lately associated with contaminated fruits and vegetables. I think one of the most recent one that comes to mind is contaminated cantaloupe. This guy can survive and thrive in pretty cold temperatures. So it's kind of associated with lunch meat as well. So if it's in a fridge, usually for most organisms, we put food in the fridge because it slows bacterial growth. Listeria can grow just fine at colder temperatures, so you can have a lot of bacterial overgrowth and get sick from that. Listeria is usually self-limiting with the infection unless you're pregnant. Listeria has the ability to cause the loss of the baby, so we're pretty careful with Listeria, especially around pregnant mothers. Non-hemolytic colonies are sometimes referred to as gamma hemolytic, even though they're not hemolytic. That is kind of the third category. So here you can see gamma hemolytic, which means that they aren't hemolytic. It's just normal growth on a normal surface. Alpha would look like this, so it kind of looks green in color. Sometimes the media surrounding it will also look a little green. And then beta hemolysis is complete hemolysis. You can see that it's completely eaten through the blood cells, and there's a halo around it. If I were to put this up to a light source, you would see like a yellow glow around the bacterial growth. Size also helps us make a determination of presumptive identification. So most colonies can be described as being large, medium, small, or pinpoint. And those sizes are kind of subjective. Pinpoint is literally if you were to take a pencil and put a little dot on a table, that's pinpoint. Small would be a little bit bigger than that. Medium would be like dime-sized. And large would be anything greater than like the size of a dime. Or, medium would be bigger than an eraser and up to a dime size. And large would be anything greater than a dime. So that kind of gives you an idea. Most of the times we do not find large colonies when we're culturing bacteria. Large colonies would usually be found if we're culturing some type of fungus. It helps suggest what you might find on the gram stain. Gram-positive bacteria are generally smaller than gram-negative bacteria. Staphylococcus is usually larger than Streptococcus. Streptococcus is kind of tough to grow. It's a little bit tricky. You have to put it in 5% CO2, otherwise it's not usually happy. It is aerotolerant, and it's tough to grow large quantities of it. So here you can see the size comparison between gram-positive and gram-negative. Of course, there's exceptions, but in general, the gram-positives are going to be more of the pinpoint bacteria, while the gram-negatives are going to spread out more and grow thicker on the plates. The form or margin refers to the edge of the colonies, smooth, filamentous, rough, or rhizoid. Examples would be Bacillus anthracis is filamentous. Diphtheroid colonies have rough edges. You can see swarming or a hazy blanket of growth on the surface. Proteus does swarming. When you get Proteus in a lab, it's kind of a pain in the butt. So we have to grow it by itself if we suspect that it's Proteus, because Proteus will literally swim over and take over the entire plate. When we do certain antibiotic susceptibility testing, we always do a purity plate to make sure that the culture that we're growing is pure before we evaluate whether it's susceptible or resistant to an antibiotic. And if you have a Proteus species, it has to go on its own purity plate. You can never put it with other organisms because it will swim all over the plate. Dry appearance, diphtheroids, so gram-positive bacilli. Yeast is a creamy white to yellowish with little feet or pedicules. So here you can see some common shapes of the form or margin. And here is Proteus. So Proteus was initially plated in the middle, and then it kind of swims in these waves over the entire plate. So this entire surface is swarmed with Proteus. This was a red plate, and because Proteus is growing on it, it's swarmed over the whole plate. It's now like a brown in color. Here you can see an example of diphtheroid colonies. And then the elevation, determined by tilting the culture plate and looking at the side of the colony. You can see raised, convex, flat, umblicate, or umbinate. Raised has a raised and flat top. Convex is dome-shaped. Flat is not raised. Umbilicate, convex with a depressed center, or pitting. S. pneumoniae, if it doesn't have a capsule. And umbinate, complex or convex with protruding nipple. Diphtheroids would have that. So you can see this right here is an example of what that would look like. And that's umbinate. Here are the illustrations: flat, raised, convex, umbilicate, and umbinate. Colony density may be described as transparent, translucent, or opaque. Beta hemolytic strep is translucent. Group B strep is semi-opaque, and most staph and gram-negative rods are opaque. As we're going through this class, you're going to learn some specific details about different microorganisms. So up front, I kind of present everything. You really want to focus on the study guides to make sure that you're studying what you need to know for now. You don't have to memorize everything up front. By the end of the class, all of these little details you will know and you will have learned. But you want to kind of pace yourself and not over-memorize up front, because there's a lot of information here. So pay attention to the things that are on the study guide and the things that you're supposed to know now. And then as we start to learn more and get deeper into the course content, by the end of the course you will know these details. Color is used to describe a particular colony, genus in general. So white, gray, yellow, buff. Different colony colors are important to note when we are working up culture plates. Coagulase negative staph are white, Enterococcus and most gram-negative rods are gray. Micrococcus and Neisseria are yellow or off-white, and diphtheroids are buff. You can see different examples of colony color. You can see how white those are compared to these. Consistency is determined by touching a colony with a sterile loop. Brittle, it splinters. Creamy, dry, waxy, or sticky. Sticky, the entire colony comes off the plate. So Staph aureus is creamy, Neisseria is sticky, Nocardia is brittle, crumbly, wrinkled, Streptococci is dry, except for the mucoid varieties, and Diphtheroid are dry and waxy. Pigment, inherent characteristic of specific organisms. The pigment would seep into the agar. So it wouldn't just be on the actual organism. If it's pigmented, it'll seep into the agar. So P. aeruginosa, Pseudomonas aeruginosa, has a green or green metallic sheen. Serratia marcescens is red. Chloe vera is blue. Chromobacterium violaceum is purple. And Prevotella is brown or black anaerobe. So you can see some examples of the pigment. And you can see what I mean by how it seeps into the agar. Odor. There are distinctive colony odors that help identify organisms. Common practice used to be like wafting the smell of the plate, and some microbiologists still do this. Some labs are now saying don't do that. If you're pretty certain that there's no spore formers on the plate, you can still practice wafting. You can still try to smell the plate to see what it smells like. Smell is not something that would typically go in a report. It would just give you, the microbiologist, more assurance of what you're working up. Staph aureus smells like an old sock, kind of moldy, nasty, gross. P. aeruginosa, they say, smells fruity or grape-like. I have smelt different types of P. aeruginosa, and it doesn't always smell like grapes, but if you get that grape-like smell and you're looking at a plate that's got the green like halo around it and it looks kind of gunky and mucusy, you can safely assume it's P. aeruginosa. Proteus mirabilis is putrid. It smells like death. Haemophilus smells like a musty basement, and Nocardia smells like freshly plowed field. Different types of organisms are going to take on a different type of scent, but that is not necessarily a diagnostic note that we would put in a medical record. Colonies with multiple characteristics. So here you can see these are kind of seeping. They have this uneven edge. They look kind of powdery. Here you can see there's some pitting into the agar. Streamers, vine-like growth in the media or puffball-like, so Streptococci. Scum-like growth, yeast, and Pseudomonas species. Turbidity, the overall cloudiness of the liquid, and if there's gas bubbles present, enterics would be indicated. So here you can see examples of growth in liquid media. Here's like that puffball. Here you can see it's an obligate aerobe. It needs that oxygen, so it's going to be up toward the oxygen surface. Here you can see some flocculent growth or air bubbles in here. And then here you can see this is likely a Pseudomonas because it looks really gunky and has a film on the top. And here's a couple other examples. This would likely be an obligate anaerobe because it's hiding underneath in the away from the oxygen. Strep pneumoniae versus viridans. So both of these have the potential to have that the alpha hemolysis where we have the green-like growth. But you can tell the difference in the colony. So strep pneumo is umbinate, while viridans has an umbinate center. So it has that, or umbilicate versus umbinate. So umbilicate means that it kind of bows into the agar a bit, while umbinate means that it pops up in the center. S. pyogenes versus S. algalactiae. So here you can see the colonies and how it's a little bit different. So S. pyogenes has a lot more beta hemolysis. You can see it very obviously. And then you can see Strep algalactiae also has beta hemolysis, but the zone of hemolysis is much smaller, so it's harder to see. And then Staphylococcus organisms versus yeast. So one way that you can tell it's a yeast is it looks like star shapes. Like it looks like there's one central cluster, and then there's these little tiny star-shaped arms that are coming off of it. While bacteria is usually perfectly circular, or it has like a more definitive shape. So if you see bacteria that kind of have this convex growth to it, you see the growth and you kind of see these projections that are coming off of it, it may mean that it's a yeast growing.